Thank you very much. I'm going to introduce myself, but I don't have to now. I am in the Department of Genetics. I apologize if I stop talking about genetics. I'm going to try and sort of define everything that's relevant. So, half of my work is really, I would say, kind of classical medical genetics, looking for genetic variants that are associated with disease or other complex traits in living people. But what I'm going to talk about today is the other side of my work, which is using techniques from statistical genetics to try and understand human evolution. Okay? And in particular, to try and understand the relationship between human evolution and environment, both the natural environment and the constructed environment, and how that's changed over pretty recent human history. I'm going to talk about two projects, and I'm going to mostly be talking about Europe, in fact, mostly about Britain. That's not because Britain is the most interesting place in the world. It is, but that's not why I'm talking about it. It's actually, just because that's where we really have a lot of data, both kind of archaeological and, as you'll see, ancient DNA data. But of course, these approaches are generalizable as well. So I'm going to talk about two projects, one looking at natural selection over the last 5,000 years, and we'll talk about why that's an interesting time period, and then another project actually looking at even more recent selection, in fact, natural selection in living generations, so over the last 50 years, and then what we can learn about the role of culture in natural selection and genetic effects by comparing these things. But a bit of background for people who aren't so familiar with history. So if we think about the history of Northern Europe, and Britain in particular, modern humans have lived in Europe for something on the order of 45,000 years. Okay, so before that, there were Neanderthals, other archaic humans, but modern humans, what we think of as anatomically modern humans, the ancestors of everyone alive today, about 45,000 years. And, it's sparse, but there's a pretty good record of culture from sort of artifacts and cave paintings and things. And, I think obviously there's kind of variation, but we do get, we get a sense of a kind of long standing way of life. So you can see there are depictions of mammoths, wild aurochs that people were hunting. And we often sort of label all these people together as like hunter-gatherers. Maybe sort of like a little bit kind of crude, but that is the lifestyle they were practicing, a lifestyle based on the consumption of wild resources. And that would have been familiar, for tens of thousands of years, and it probably would have been very familiar to people like Neanderthals who were living there before. And then this is the sort of environment that these people inhabit. So this is the one that we named Iain Gorges, the Danube River in southeastern Europe, and you can see there are sort of forests, and there are this river is or was full of giant catfish, which are very delicious to eat. And so it is the kind of natural environment that people inhabited. And then, starting about 8,000 years ago, that natural environment starts to change. And it starts to change because humans start to change. So the major transition, possibly, arguably, the major transition in all of human history is the development of agriculture, starts in what's present-day Anatolia and spreads into Europe, starting about 8,000 years ago. Okay. And that transition is accompanied by sort of dramatic cultural change as well. So we see pottery, which didn't exist before, domesticated crops and animals, of course. We start to see larger, more permanent settlements. This is like a early Neolithic house, didn't really exist before. And really, it's hard to kind of overstate overall just how much of a shift in lifestyle this involved. And then, of course, things continue to change. So as we go into the Bronze Age, starting maybe 4,000 or 5,000 years ago, we see domestication of horses, the development of wheeled vehicles, the spread of new kind of cultural innovations, for example, Indo-European languages, distribution of Indo-European languages as they are spoken today, really remarkable. In fact, the majority of people in the world speak a language which is from this relatively small language family, which probably started spreading out 4,000 or 5,000 years ago. We also see the development of kind of social changes as well. So, for example, this is a skeleton of a man whose genome we sequenced a couple of years ago. He is from a place called Varna in what's present-day Bulgaria, and he was basically the richest man in the world, like 5,000 years ago. Okay? So this is the first burial. You can sort of just about see the kind of gold ornaments here. this is the first time where we start to see sort of a hierarchy among variables. Okay? And so that's kind of associated with social status. Okay, and from the archaeology, we have a pretty good record of the spread of these kind of cultural innovations. So this is a map of the spread of agriculture throughout Europe based on archaeological data. You can see the earliest Western Eurasia. You can see the earliest agriculture is sort of here in southwest Anatolia, southeast Anatolia, sorry. And it spreads out through Europe. These are dates BCE. And within a few thousand years, it's reached really the extremes of Europe in Iberia, Britain, and Scandinavia. So we know, so we actually have a pretty good idea of the timing of the spread of these sort of cultural changes. And of course, the question that sort of plagued archaeology for a long time was, are these cultural changes associated with the movements of people? Okay, so is it just that people in Europe are sort of learning to farm and then you see your neighbor farming and you decide it's cultural transmission, or does this actually represent the movement of people, migrations, massive movement? And this debate sort of went back and forth, of course, but really there's no way to infer that without looking at people. And the first person to really kind of try, people thought about this before, the first person to really try and do this by looking at the genomes of living people was this man, Luigi Luca Cavalli-Sforza. And this was really in the 70s. And so he sort of did analysis like this. So this is a map of Europe showing principal components, essentially gradients of gene frequencies across Europe. And so, and Cavalli-Sforza and his colleagues saw these kind of gradients of gene frequency. So it's sort of start, high in Anatolia, and it gets lower as you go throughout Europe in this kind of north-south direction. And they saw this as evidence for migrations of people. So they said that you could see the shadows of these ancient migrations in the genomes of living people. So that was very sort of inspirational, but unfortunately, it turns out that that's not really true. Or at least there are other models which generate, turns out there are other models which generate the same patterns. And you can't actually infer the genome, you can't actually infer the movements of ancient people by looking at the genomes of living people. I think that's sort of like pretty widely accepted now. So what can you do instead? You can try and look at the genomes of ancient people, okay? And so this is the field of ancient DNA. And, people started trying to do this in the 80s, but for various reasons, never really worked. And it was only really in around 2010 that it was actually possible to do this, to sequence the genomes from ancient individuals. So this is the original publication of the Neanderthal genome. These are Neanderthal remains from a cave in Croatia, about 45,000 years old. And this was really the first time that anyone had actually managed to sequence ancient DNA. You may have seen the Nobel Prize in Medicine was awarded to Santi Tabo for essentially this work this year. Now, Santi and his colleagues have focused mostly on Neanderthals and other archaic humans, but actually you can also use this to look at the genomes of people who lived at the time that we're talking about. So development of farming and the Bronze Age. So here's an example. You get one picture from the lab. This is a tooth from someone who lived in Britain about 3,000 years ago, and we're being very careful in the clean room and to cut off the end of the tooth and drill into the middle to try and extract its DNA, which is not contaminated by contact, and so on. And then we're able essentially to sequence the genome of this individual. And so that allows us to actually look at genetic variation changing over time. Okay, so we can actually now really ask the question of whether these movements are the result of changes in movements of people or culture. And it turns out that they are largely associated with movements of people. Okay, so I'm going to talk you through one way in which you can see this. So here is a map of principal components of genetic data from about 1,000 ancient people. So each dot is an ancient individual. And you can think of this principal component analysis as just a sort of summary of their genetic data. So the distance, essentially the distance between two individuals on this plot is proportional to their genetic difference between their genes. And the colored points are ancient individuals, that colored according to this timeline here, going back to 45,000 years ago, up to about 1,000 years ago. And the grey points here are people from present-day Europe. And so these are people from Northern Europe and these are people from Southern Europe. So the first thing you can see is that the vast majority of the people who live in Europe over the past 45,000 years are actually genetically nothing like people who live in Europe today. Look at the oldest individuals here, they're sort of in the middle. This is basically going through the Ice Age down to about 10,000 years ago, and then the sort of large hunter-gatherer group in the Mesolithic, they're sort of over here on this side of the plot, and then really very tightly associated with the introduction of farming, we see a movement over here. And just to give you a sort of a sense of what this distance means, the genetic distance between these people and these people is sort of similar to the genetic distance you would see between present-day Europeans and present-day East Asians. So these are really very like highly differentiated populations, and really completely sort of replacing people who lived there before. So there is some mix. And then in the Bronze Age, we see additional movements of people, and then finally, about 4,000 years ago, we start to see people who look like present-day Europeans. So this really, answers the question that Cavalli Sforza was trying to answer and show us that really these sort of major cultural transitions, at least in Europe, were associated with movements. And actually, because we know where these individuals came from, we were able to actually reconstruct the geographic movements. And we know the location of each of these individuals, so we're able to reconstruct the geographic movement as well. This is one of the classic early papers doing this. This is showing the direction of migration of people from Anatolia, early farmers from Anatolia into Central. That's just love drawing big arrows. Bronze Age migration, that sort of seems to originate here in the Pontic-Caspian steppe. There's this group called the Yanaya that actually turned out it's one of the original groups to domesticate horses, which is probably why they were able to move so fast. Okay, so that's kind of cool. So we're able to draw big arrows and maps and talk about large-scale migrations. Now, there's actually tens of thousands of ancient genomes available. And so now we start to have really dense sampling, and we're able to ask more kind of focused questions, really about sort of fine-scale social organization. So I'm just showing you here a couple of studies. So this is a study of central Germany, where they're actually, by sequencing the genomes of people who are buried in different cemeteries, they're actually able to reconstruct the familial relationships and the relationships between the different cemeteries, and start to actually ask questions about how social organization, how societies were organized in these different populations. So we can actually really now have very sort of fine-scale information about large numbers of individuals. Okay, that's kind of exciting. That's not what I'm interested in. What I'm really interested in is asking the question, biologically, how did human populations adapt to all of these changes? So what are the biological changes? What are the genetic changes that accompany these cultural and environmental shifts? And how do they relate to genetic variation and phenotypic variation today? Okay, so what can we say about our own genomes based on understanding how we evolved? And we started this project kind of a long time ago, and this is sort of the first paper we published looking in 2015, looking for signals of natural selection in the genome over the past sort of 8,000 years. Okay, so this is really the time period encompassing the agricultural transition and the Bronze Age transition. Okay, so I'll show you a view of this map. This is all a Manhattan plot because it's supposed to look like skyscrapers. So on the x-axis is a map of your genome. So you have 22 chromosomes, or even sex chromosomes, and each point here is a single genetic variant, SNP. And they're sort of organized along the chromosomes. And the y-axis here is a measure of how much that SNP fits overall patterns of genetic variation in this population. Okay. And when you see a SNP that's kind of up here, that means that SNP does not fit the genome-wide patterns. Okay. So it's an outlier, and we interpret those outliers as being the result of natural selection. Okay. So when you see a tower here, it means there's a lot of different genetic variants which are kind of out of the null distribution. And so we interpret that as meaning that that part of the genome, that could be a particular gene or it could be some non-coding region, has been the target of natural selection. And I'm labeling each of these peaks with the names of the genes that are in these targets. That may not sort of mean anything to you, but actually it turns out that all of these genes are fairly well characterized, and we actually understand the function of most of these genes. So if we go and ask what they do, it turns out that they fall into really three main categories. So there are genes associated with diet, genes associated with skin pigmentation, and genes associated with the immune system. And that kind of makes sense, right? So we know that diets are changing dramatically in this time period. It's sort of reasonable that we might see adaptation to diet. We'll talk a little bit more about skin pigmentation, but in particular, people are moving into northern latitudes where there's less sunlight. So we expect to see selection for lighter skin pigmentation. It's a little bit more complicated, as we'll see. And then people are moving first into a new environment where there might be different pathogens, but actually, perhaps more importantly, are moving into, now have domesticated animals and living in close proximity with domesticated animals. So we expect to see sort of zoonosis and a different kind of immune environment. So this is kind of, as a big picture, this is kind of the sorts of things we might sort of expect to see. Okay. But as I said, this is really looking at natural selection over the last 8,000 years. It's a little bit kind of non-specific, and we don't know, which of these things are related to sort of particular transitions, particular kind of cultural changes. So that's really the motivation of our most recent study, which is to look more specifically at a more specific time and place and see whether we could actually kind of say anything about what's driving this natural selection. Okay, and so this is work with Jonathan Turvos, who's in the Department of Statistics in Michigan. And basically, the punchline here is that we show that, at least in Britain, over the last 5,000 years, vitamin D is really the only thing driving natural selection. It's by far the strongest selective pressure. And I'll talk about how. Okay, so these are the data we look at. So these are we're looking at data from Britain. These are ancient. These are the sites where these individuals come from across Britain. This is a histogram of the dates of each individual. So you see it's sort of quite non-uniform, but we do have sampling going back at least 4,500 years. And we stop at 4,500 years ago because this is the last sort of major transition in ancestry. So all of these people are sort of fairly homogeneous in terms of genetic ancestry. So you can see this here on the PCA plot. So again, this is a sort of summary of genome-wide genetic variation. The gray points are present-day individuals from Western Eurasia and North Africa, and you can see sort of genetic, you can see sort of geographic structure. So Northern Europeans here, Southern Europeans, and then the Caucasus and down to North Africa. And these light blue points are present-day British people, and these dark blue points are these ancient people from the last 5,000 years. Okay? So you can see that they're sort of genetically clustered very tightly together. We can treat this as more or less as a single continuous population. Just for comparison, if we look in Italy, we can't do that. So, for example, here are ancient individuals. Here are present-day Italians, and here are ancient individuals from the same time period from Italy, and you can see there's much more variation in ancestry, and we can't really treat this as a single population. Much more migration around the Mediterranean. Okay, so then what we do is we ask, if we look at how if you look at all the variants that we've detected in the genome, and we ask how fast they change over time, what does that look like? So here's the same kind of plot. So again, this is the log p-value. So the null, the null hypothesis here is that the allele frequency hasn't changed over time. It's just kind of drifted randomly due to genetic drift. And the alternative hypothesis that we're testing here is that there's been some systematic change in allele frequency. Okay, and so we identify these 5 regions. This is kind of an artifact, and actually these are all sort of genes that we kind of know the function of. Okay. If you look at these, it's not sort of immediately obvious that they should share a common function, but it turns out they actually do. So the strongest signal here is actually the strongest signal of selection in the entire human genome in any population is on lactase persistence. So this, as people may know, most people in the world are able to digest lactose, the main sugar in milk, as children, and then lose that ability sometime during kind of early teenage years. Okay. And in some parts of the world, in Northern Europe, in the Middle East, and in parts of Africa, it's relatively common for people to actually, for that ability to persist into adulthood, okay, to be able to digest lactose. And that's actually controlled by a single genetic mutation, different mutation in different parts of the world, but in Northern Europe, it's one particular genetic mutation, and that has been very strongly selected over the past 5,000 years. We see variants associated with skin pigmentation, variants associated with vitamin D metabolism, and then this region here that I've laid is a sort of kind of special region of the genome that contains a lot of genes associated with the immune system. If we look at what these signals are actually doing, there's kind of 3 independent signals here associated with and largely associated with autoimmune disease, particularly celiac disease. Okay, so what's the hypothesis? So actually, this is an old idea for what the selection lactase persistence, which is that it's related to vitamin D metabolism. Okay. So the idea is, you synthesize vitamin D from sunlight in skin. Okay. And one of the major functions of vitamin D is to allow you to absorb calcium, right? Which is obviously important for bone development. Okay. And the idea of selection lactase persistence is that it allows you to consume milk, which milk contains a lot of calcium, and it contains a small amount of vitamin D, but enough vitamin D to allow you to digest the calcium that's present in milk. Okay. So one hypothesis is that that's what's driving selection on lactase persistence. Now it turns out that selection for lighter skin pigmentation also increases vitamin D production. Vitamin D, this gene BHCR7 is directly involved with vitamin D metabolism. And it's not so obvious what these kind of autoimmune traits should be, but actually it turns out that celiac disease, even today, is a major risk factor for low vitamin D and osteoporosis. Because if you have celiac disease, you have an inflamed gut, you can't absorb as vitamin D as easily. So actually, a kind of parsimonious explanation for all of these observations is that the thing that's driving natural selection here is a lack of vitamin D. Now, I can tell you that one of the distinguishing features of Britain is that there is not a lot of sunlight. And so the hypothesis would be then, as people move into Britain and transition to a diet which is like very heavy on agricultural produce, they then have extreme vitamin D deficiency. Okay? And so we see selection on all of these kind of different pathways increase your amount of vitamin D. Again, we see how cultural and environmental changes are what's driving genetic adaptation. You might reasonably ask, okay, so it's cloudy today, it was cloudy 5,000 years ago. Why did people who lived there before not have this issue? Because it turns out they were eating a very different diet. So unfortunately, in this figure, the classic paper, unfortunately, this figure time runs backwards. So this is the past. These are hunter-gatherers. These are the farmers. And this is a measure of carbon stable isotopes in the bones of these individuals, and sort of the interpretation here is kind of a measure of diet. And the point is basically that the farmers are eating a diet that's based very heavily on terrestrial products, whereas the hunter-gatherers actually are eating a lot of fish, which is naturally very high in vitamin D. So probably they don't have this issue because they can actually absorb enough vitamin D from their diet. So again, this really sort of emphasizes how cultural change in terms of the development of agriculture drove biological natural selection and actually things that we still observe today, for example, variation in risk in autoimmune disease. Okay, and we can actually see even more fine-scale changes. So here on the right, so looking at these kind of top loci here on the right is the estimated allele frequency. So this would be the present day, this would be 4,500 years ago. The blue line, for example, shows how we estimate that the frequency of the lactase persistence allele has gone from essentially 0 to 60% today. And on the left, this is an estimate of the strength of selection. So we'll talk more about this in a minute. But essentially, this number is proportionally how many surviving children you expect to have if you carry this allele. Okay. And so you can see that we actually estimate that the strength of selection changes. So for the advantage, if like 150 generations ago, if you carried the allele, you expected to have 8% more children, which is an enormous effect. But then that seems to change, that seems to decrease over time, and the advantage of these other alleles seems to increase. Okay. So there's a kind of time period in the middle here where we sort of see shifts in the strength of selection on these different alleles. And that's potentially evidence that there are cultural shifts happening at this time that actually have changed. So actually now, people, now perhaps it's not so good to have lactase persistence, but actually it's better to have lights of superpigmentation or more vitamin D tablets. So even on these kind of fine scales, on the time scales of tens of generations, we see changes in really what is driving the selection there. So that sort of got me thinking. So we look at these these plots, and we're thinking of like changes over thousands of years, and these people just sort of like drifting on kind of tides of history. But what did it feel like to actually be there in that time? What did it feel like if you were sort of experiencing natural selection here? So we decided to try and understand that by looking at really recent natural selection. So this is work with Melinda Mills and John Perry. And the idea here is that in order to look at natural selection in recent times, what we're going to do is a genome-wide association study for fertility. I'll talk about what that is. So the phenotype here is the number of children, like the number of children you've ever had. And the idea is that if there are genetic variants associated with fertility, by definition, those genetic variants are under natural selection. Okay. And conversely, presumably the main way in which natural selection works on living populations is through fertility. Okay. Again, I'm talking mostly about Northern Europe. Most children survive to adulthood. So number of children is a pretty good measure of the number of surviving. Okay, so what is a GWAS? What is genome-wide association study? Just in case people aren't aware of this. So this is kind of a very naive picture. So let's suppose I want to test whether a particular genetic variant is associated with phenotype. Let's say height. What I'm going to do is collect data from a bunch of individuals. Each of these points is a person. I ask how many copies of the variant do they have. You either have 0, 1, or 2 copies. And I regress that against the phenotypic value. And if I see a slope here, is a measure of the effect size of that genetic variant. And the genome-wide part is that I'm going to do this again across the genome, each point of the genetic variant for all of the, or a large number of genetic variants in the genome. So it's sort of an unbiased scan. And this is, for example, a genome-wide association study from Height in the UK Biobank. And there are actually thousands of genome-wide significant variants associated with Height. So this red line is significance level corrected for multiple tests. That's genome-wide association study. And people have done these for, I imagine you've seen these in other talks. People have done these for like pretty much every trait you can think of. Okay, so why is it a bad idea to do it for fertility? So there are sort of like the obvious bad idea, which you might think, is that, well, but fertility is complicated and people have children for all sorts of reasons, and there are all kinds of other factors that go into whether you have children. So how can you like measure genetic effect? That's actually not the biggest problem. I mean, not that it's not a problem, but it's also a problem for all other phenotypes as well. Okay. Same argument if you do a GWAS for heart disease or height. So there are, I'm not going to go into the details, but there are ways of dealing with that. There's actually a more fundamental problem, which is that if you imagine a genetic variant which increases fertility, it's rapidly going to increase its frequency through the population because it increases fertility, it's under selection, and pretty soon everyone will carry it. Conversely, a genetic variant which decreases fertility, which makes you infertile, isn't going to last for very long. So we don't expect to see much genetic variation in fertility. Okay, so sort of geneticists would say that the genetic architecture is dominated by negative selection. We expect the heritability to be very low. We expect it to be dominated by very small effects of very rare variants, which are statistically difficult to detect. And in fact, that's borne out empirically. Here's a study looking at a bunch of different phenotypes on the x-axis, and the log scale is sort of the number of common variants associated with the trait. The y-axis is the number of rare variants, and the number of children is much higher than any other disease phenotype. So it's just statistically very difficult to detect that. So the only thing that you can do is to then make a very big study. Okay, so we've collected a bunch of different cohorts together, and we've ended up with about 780,000 people. So these are all from sort of Northern Europe, but this is a sample size that we think is powered to detect genetic effects. And in fact, we do find genetic effects. So here again, it's another Manhattan plot, x-axis chromosomal position. Each of these towers is a genetic variant gene which is significantly associated with the phenotype. The two largest effects we find by a long way are these genes, CADM2 and ESR1. And the effect size here is in units of the number of children. Okay, so if you carry the fertility increasing variant of this gene, you expect to have 2.5% more children than someone who doesn't carry that variant. It's a pretty big effect. Okay, what do these genes do? Well, it turns out we know what they do. So CADM2, in fact, is one of the only, in fact, probably the only gene which is sort of robustly associated with behavioral traits, in particular risk-taking. So in this case, the allele which increases your risk-taking behavior, so this is measured by things like you drive faster than the speed limit, or like how much money do you have in your 401k, stuff like that. So the allele that increases risk-taking behavior actually increases your number of children. And the other gene is the estrogen receptor, or one of the estrogen receptors. That actually has a direct effect on reproductive biology, but also actually has a very big effect on behavior. So in mice, this kind of affects fertility by changing behavior. And the interesting thing is that both of these genes have strong signals of what we call balancing selection, which suggests that actually at some times or in some environments, they might be advantageous, and in other environments, they might not be. Okay, so that sort of explains why these relatively large effects are able to persist in the population at intermediate frequency. Right now, this effect it's advantageous to, in terms of fitness, to take risk, but in the past that might not have been the case, or in different environments that might have. And if we look at the other phenotypes, then we see sort of a wide range of associations. Sorry, if we look at the other associations, we see a wide range of associations with other kind of behavioral and reproductive related traits. So here, each column is one of the fertility increasing variants, and we're showing the z-score of association with these other phenotypes. So you can see that some of them seem to act through things like testosterone and sex hormone levels, other than act through behavior, age, sex, and so on. Okay, so actually kind of disentangling what the kind of causal effects of all these genetic variants is not easy. Okay. So those are kind of interesting, but actually when I saw these results, the thing that I was most interested in, not those genes, but this gene called FADS1. So that has an effect which is about half the size of CADM2 and ESR1. And the reason why I was interested in this is because this is the only one of these genes which is under selection today, which was also under historical selection. Okay. So this is kind of interesting because it's the only gene where it's been under selection for in the past sort of 10,000 years, and it still seems to be under selection today. So that seems like it might speak to something kind of fundamental. Okay, so what does this gene actually do? Well, it turns out that we know what it does. So each bubble here is a particular sort of fatty acid. On the right hand here, you have long chain fatty acids. This first number is the number of carbon atoms in the chain. And on the left hand here, you have short chain fatty acids. And these long chain fatty acids are important for cell membrane development, particularly brain development, actually important for many things. Okay. And if you eat a diet which is high in plant fats are largely sort of short chain unsaturated fatty acids. So you get a lot of these. And you need to synthesize these long chain fatty acids. And this gene FAD1 and the related gene FAD2 actually catalyze this process. Okay. On the other hand, if you eat a diet which is very high in meat or marine fats, you get an input of longer chain fatty acids, so you don't then need to have as much enzymatic activity. Okay, so actually we know exactly what this gene is. And it turns out that selection actually has been pretty consistent over the last 5,000 years. Okay, so if we look at the frequency of allele which increases enzymatic synthesis in different parts of Britain. This is sort of directly inferred from ancient DNA. You can see the frequency has now increased consistently by about 0.008. It's not sort of going up very fast, but it's consistent. And somewhat remarkably, if we look at UK Biobank as a function of birth year, it increases by actually almost exactly the same amount. So again, this effect seems to be, whatever it is, it's been consistent over the last 5,000 years. It seems to be more or less consistent over the last 50 years as well. And it turns out that, in fact, it's not just the last 5,000 years. This variant has been under selection in different ways for hundreds of thousands of years. So here, I'll talk you through this in a minute, but here's a tree relating present-day human populations. So in Africa, in East Asia, Native Americans, and Europeans. This is sort of the relationship between these major population groups. And what I've done, what's been sort of reconstructed through a large number of studies, is taking the evolutionary history of this variant. So I kind of colored in the tree as to whether the ancestral or derived variant is more prevalent at different times in this history. What you can see is most recently, say in Europe, the derived variant, that's the one that increases enzymatic activity, has been common and increased in frequency, but actually at other times in the past, the blue variant has been more common. And it turns out we can relate these changes to changes in diet. Okay, so this is what the distribution looks like in present-day populations. Actually, again, the derived allele is in red, and the ancestral allele is in blue. This grey allele is actually a kind of derived allele. And so what's interesting is that actually in Africa, in present-day African populations, we only see the derived allele. We never see the ancestral allele. And because the splits among present-day African populations are actually deeper than the splits among African and non-African populations, that tells us that the derived allele must have become fixed in Africa before the split of African and non-African populations. We estimate, it's a bit hard to estimate this date, but on the back of the envelope, not back of the envelope, using a method which has a very high uncertainty, we estimate this is something like 300,000 years ago. Okay. That's sort of consistent with, say, the development of fire and the use of kind of things like a transition in diet that involves like the use of tubers as a major food source. Okay. So like it's kind of like this may plausibly be associated with a transition to a more kind of plant-based diet. Then if we look at then the Out of Africa event, which happens here maybe about 60 or 70,000 years ago, split of present-day non-African and African populations. If we look at early non-African populations, we see that actually almost everyone carries the ancestral allele. Okay, so even though the red allele, the derived allele, was kind of fixed in the ancestral African population, in non-Africa, in early non-Africans, everyone's carrying the ancestral allele, and that's. If we think about the diet of these individuals, that's kind of consistent. These guys are like the mammoth hunters, right? So once people move out of Africa into Eurasia, they encounter all these megafauna, mammoth, that are not used to humans, and we can tell, again, by looking at stable isotope values, that their diet almost entirely consisted of meat products. And so that we would propose that actually then that would entail selection for this less enzymatic activity allele. And then, of course, so you actually see a switch. We actually see a switch back here just after the Out of Africa event. And then, of course, in more recent times, after the development of agriculture, independently in both East Asia and Europe, we see selection for the increased frequency. So that's this kind of nice part. Okay. And for example, present-day Native American populations, split off before the development of agriculture, retain the ancestral. So actually using HDNA and by combining this information with information about diet history, we're able to kind of reconstruct this trajectory. Okay. So it seems like this allele kind of acts as a sort of tuning switch, allowing people to adapt to different types of diet. Why? We don't know. Okay, so what is actually the selective pressure? That's what we still don't know. However, a clue comes from work in mice. So actually, if you knock out FADS2, which essentially works in the same pathway in mice, then both male and female mice become completely infertile. But you can actually rescue that phenotype by feeding them diets that are high in long-chain unsaturated fatty acids. So it seems like at least we have a good model in mice, which is kind of consistent with what we've seen in humans to try and understand what's driving this natural selection. And so we're following up with these mice, trying to understand actually the biology of what's going on. Okay. It's just overall, this is I think, an example of how, even over very long timescales, changes in human culture and environment can affect sort of biological evolution. And for example, today, this allele is actually most known because it's strongly associated with cholesterol and heart disease. Okay, so again, this kind of allele, this association with diet and these kind of selective pressures are not just affecting kind of evolutionary variation, but also affecting medically and functionally relevant variation. Okay, final thought. So I looked at, when I looked at this, the only gene that was actually sort of selected in the past and in the present was FADS1. Okay, so all of these things which are apparently under selection today weren't under selection in the past. And all of the things that were under selection in the past, lactase persistence, skin pigmentation, so on, weren't under selection today. You can sort of summarize that. So here, these are, again, some Manhattan plots showing selection over different timescales. Okay, so these are sort of different statistical approaches to look at selection. This detects selection in the last 50,000 years, the last 10,000 years, the last 2,000 years, and the last 50 years. And just the sort of takeaway here is that, again, we sort of show this statistically, but there's very little overlap across these. The only, lactase kind of leads across multiple timescales, but in fact, the only signal in the entire genome that kind of jumps across different timescales is FADS1. And statistically, there's no more overlap between these than you expect by random chance. Okay. So what that tells us is that the evolutionary pressures, at least even in sort of recent human history, have changed very rapidly over time. And we've seen, even on the timescales of hundreds of generations or tens of generations, we've seen rapid change. And now on the timescales of thousands of generations, tens of thousands of years, we see the same kind of rapid change. So that tells us that if the evolutionary pressures on these genetic variants are changing, then and are sensitive to changes in environment and culture, then the effects of those genetic variants are also changing and are also sensitive to changes in environment and culture. And if they're sensitive to changes in environment and culture over time, they must also be sensitive to differences in environment and culture today. And I think that is how we should really start to think about genetic effects. And you would have seen, a lot of people think about the results of GWAS now in the context of either predicting disease and medical genetics, or even in the context of sort of social phenotypes. Okay. But in fact, we know, at least from thinking about the kind of evolution of these variants, that their effects cannot be consistent over environments. So I think that that's something that's really missing, both on the kind of medical field and in the sort of sociogenomics field, is actually a real kind of like understanding and modeling of how these effects vary over context. Okay, that's all I wanted to say. I would thank my lab and collaborators elsewhere, and of course many postdoc positions available if anyone's interested in thinking about this. And I'd also like to thank all my funding sources, particularly the NIH and NSF, who funded most of this work. And I'd like to thank you for your attention.